Hello students, welcome to the EPG Pathshala. My name is Dr. Rajeshwari Sinha and in today's lecture we are going to learn about proteins. We will do an in-depth study of protein classification, chemical structure and functions. Let us see what objectives we are going to address in this lecture. We are going to learn about the basic concept of proteins, the classification of proteins, to know about synthesis of proteins, their structures and techniques used for determination of the structure of proteins. We are also going to have a look at the various functions of proteins. We hope you have an enjoyable time listening to this lecture. Proteins are macromolecules present in all living organisms. They are highly complex molecules having high nutritional value and plays an important role in biological processes. The name protein was derived from the Greek word proteos, meaning holding first place. Protein is a polymer of amino acids arranged in linear chain. Polymerization of amino acid takes place via peptide bond formation. In this slide, we look at the formation of a peptide bond. Two amino acids, amino acid 1 and amino acid 2, combined with each other. The carboxyl end of amino acid 1 reacts with the amino end of the amino acid 2 to form a CONH bond along with the release of water. The CONH bond is also known as the peptide bond. All the proteins are encoded by the 20 genetically coded amino acids called standard amino acids. Their occurrence is very specific, which differs from species to species and organ to organ with different functions. Central dogma. Francis Crick in 1958 proposed the term central dogma, according to which in living beings information is travelled from DNA to RNA and then from RNA to proteins. The figure below shows the graphical representation of the central dogma. Classification of proteins. Proteins are of basically two broad types. They can be classified based on the number of peptide chains and again based on composition. According to the number of peptide chains, they can be monomeric protein having single polypeptide chain, for example myoglobin, or oligomeric protein having more than one polypeptide chain, for example hemoglobin. On the basis of composition, proteins can be simple protein composed of only amino acids, for example insulin, or they can be conjugated protein made up of amino acids and additional organic and inorganic groups called prosthetic groups, example glycoproteins and lipoproteins. Proteins can also be divided on the basis of tertiary structure. They can appear as globular proteins, fibrous proteins and membrane proteins. Globular proteins are soluble and many are enzymes, for example amylases. Fibrous proteins are often structural proteins such as collagen, connective tissues, keratin and the protein component of hairs, nails. The membrane proteins serve as receptors or channels for polar or charged molecules to pass through the cellular membrane, example GPCR. Classification of proteins. Classification can also be done on the basis of tertiary structure, complexity and number of subunits. Based on their tertiary structure, protein can be divided into globular proteins, fibrous proteins and membrane proteins. Globular proteins are soluble ones, for example enzymes. Fibrous proteins are the structural proteins and insoluble in nature, for example collagen and elastin in connective tissues, etc. Membrane proteins are generally membrane bound or transmembrane proteins which serve as receptors on cell surfaces during signal transaction or act as a channel to transport charged substances across the membrane. This shows the classification of proteins based on tertiary structure which we just discussed. We are learning about proteins today. We hope you are finding this lecture useful. So far we have understood about peptide bonds, the central dogma and classification of proteins. Now let us move forward. In the next section, we are going to see the biosynthesis and chemical synthesis of proteins and the various steps involved. We will look at the various levels of protein structure, the primary, secondary, tertiary and quaternary levels of structure, and also look at the techniques for determination of protein structure. Biosynthesis of proteins. Proteins are synthesized from the information present on the gene. It has its own specific amino acid sequence which is encoded by the nucleotides of the gene encoding the protein. The amino acids of the protein sequence are coded by specific genetic code, which is a set of 3 nucleotides known as codon. For example, AUG codes for methionine. The genetic information in DNA is coded by only 4 nucleotides, and therefore total number of possible combination of codons are 64. Therefore, there is some redundancy in codons for the amino acids, due to which some amino acids have more than one code. The genetic code of amino acid is shown in this figure. Steps involved in protein synthesis. First, DNA is transcribed into mRNA with the help of enzyme RNA polymerase. This process is known as transcription. Then, the processing of pre-mRNA takes place through the post-transcriptional modification, resulting in the formation of mature mRNA. Processed mRNA, also known as primary transcript, which is finally used by ribosome as a template to synthesize proteins. The process is known as translation. In prokaryotic protein synthesis, transcription and translation, both the process take place simultaneously. In eukaryotes, transcription takes place in the nucleus of the cell and the resulting mRNA gets translocated across the nuclear membrane, and the protein synthesis translation process takes place in the cytoplasm. Ribosomes bind with the mRNA and start reading the template with the help of anticodon located on the transfer RNA tRNA molecule. The enzyme amino acid tRNA synthetase help the tRNA by charging it with the correct anticodon. Protein synthesis always takes place from N-terminal to C-terminal. Chemical synthesis of proteins. Protein synthesis can also be done chemically by peptide synthesis methods. These methods depend upon organic synthesis methods like chemical ligation to synthesize proteins into high yield. By this method, some non-natural amino acids can also be incorporated into proteins by chemical synthesis. Through this method, we can design the fluorescent probes to attach with the protein and help in its localization, which is very useful in laboratory experiments. Drawbacks of the chemical synthesis method. It is inefficient for the synthesis of polypeptides having more than 300 amino acids. The protein structure may not get properly folded into its tertiary structure. The direction of synthesis is opposite to that of biological synthesis, that is from C terminus to N terminus. Structure of proteins. Protein structure plays an important role in its functions. Protein specificity in function is due to its unique tertiary structure known as the protein's native conformation. Proteins have 4 levels of structures: primary structure, secondary structure, tertiary structure and quaternary structure. Primary structure. Primary structure of the protein is its amino acid sequence joined together in a chain with the help of peptide bonds. Secondary structure. When the amino acid chain folds into its repeating local conformation, which is stabilized by hydrogen bonds, it is called the secondary structure. Examples are beta sheet, alpha helix, turns, etc. Tertiary structure. The 3-dimensional structure of the protein is called the tertiary structure. It is the spatial arrangement of the secondary structure with each other stabilized by hydrophobic interactions, salt bridges, disulfide bonds, hydrogen bonds, and through some post-translational modifications also. It controls the functions of the proteins. Quaternary structure. There are some proteins which are made up of more than one polypeptide chain called oligomeric proteins. The structure formed by these polypeptide chains, subunits of proteins, are called quaternary structures. The figure below gives all 4 levels of protein structures. Techniques used for structure determination of proteins. By finding the structure of protein, we can find out the structural functional relationship of any protein. To determine the structure of a protein or a protein complex, many techniques can be used. These include X-ray crystallography, NMR spectroscopy, circular dichroism, trio-electron microscopy, and dual polarization interferometry. Commonly used method is NMR spectroscopy and X-ray crystallography. These techniques provide information about the spatial arrangement of atoms in the proteins. Another method for structural determination of protein is dual polarization interferometry. It is a quantitative analytical method which determines changes in protein conformation due to any stimulus. Circular dichroism is the technique by which we find out the internal secondary structure, beta sheet and alpha helical composition present into the protein. For very large protein complexes, cryo-electron microscopy is used to provide lower resolution structural details. In some cases, like in 2D crystals of membrane proteins for higher resolution, electron crystallography can also be used. Solved structures of proteins can be freely accessible in the Protein Data Bank (PDB) in the form of their Cartesian coordinates of each atom present into the proteins. In the next section, we are going to learn about the biological function of proteins, which have cellular and structural functions and also play a critical role as enzymes. We are going to briefly touch upon the concepts of post-translational modification, protein folding, protein denaturation and energy landscape model of proteins. Biological functions of proteins. Proteins perform most of the complex biological functions and constitute most of the structural component in living beings. All the biochemical reactions in the cells are performed by proteins called enzymes, also known as biological catalysts. Enzymes are very specific, either causes formation of chemical bonds or break these bonds, resulting in the completion of the reaction. Scaffold of our body, which holds cells and organelles with each other, is made up of proteins. Cellular functions. Proteins play an important role in signal transduction also. It can transfer signals from extracellular environment to inside the cells and helps in maintaining cellular processes. Proteins can also bind with other proteins and to other smaller molecules. If a protein binds specifically with other molecules of the same substrate, it can form fibrils through oligomerization. Proteins also play an important role in cell regulation pathways through protein-protein interaction, which regulates many enzymatic activities, specifically regulate the cell cycle progression. Proteins can also act as cell receptors by getting attached with the cell membrane, act as a signal molecule also, and participates in cell-cell interactions. Proteins as enzymes. The enzymes are also known as biological catalyst, which catalyzes many biological reactions. It is proteinaceous in nature and highly specific in its functions and substrates. Most of the metabolic reactions are carried out by enzymes. It involves extensively in DNA replication, repair and transcription processes. Enzyme increases the rate of reaction by 10 to the power 17 fold. These proteins provide rigidity and stiffness to biological proteins. They are fibrous in nature, example elastin and collagen in connective tissue, keratin in hairs, nails, etc. Certain globular proteins also have structural functions such as tubulin and actin in cytoskeleton and maintains the cell size and shape. Motor protein like kinesin and myosin also have structural function in our body which generate mechanical forces and are important for the motility of the cell. Post-translational modification. Post-translational modification are of two types, one in which cleavage of bond of peptide backbone takes place and another in which addition or removal of functional groups occur in the side chain of the constituent amino acids. For the cleavage of bond, proteases are involved which recognizes the specific peptide bond associated with a particular amino acid and cleaves it, hence modifying the protein molecule irreversibly. In the other case, side chain of particular amino acid in protein is chemically modified by enzymatic action or spontaneously. For example, oxidation, glycolization, phosphorylation, etc. Protein folding. This plays a very important role in proper functioning of the proteins. Protein folding of protein is needed for the regulation of the cellular machinery and to determine the organism type. The three-dimensional structure arises due to its particular amino acid sequences in the polypeptide chain get folded and generates a compact domain having a specific structure. Specifically folded structure acts as a molecule for larger complex of proteins or forms specific site for substrate binding. Why do proteins undergo folding? The native proteins are less stable in its physiological conditions. Forces like electrostatic interactions, hydrophobic effects, hydrogen bonds, etc. act as stabilizing factor and hence lead to protein folding. Effect of hydrophobic interaction tends the nonpolar side chains to minimize its interaction with water molecules and is the important determinant of protein structure. The aggregation of these nonpolar side chain is favored by increase in entropy of water molecules. Van der Waals forces act in the interior of the molecule which are relatively weak and act for a short time. The hydrogen bonding also plays an important role since protein folding occurs in a way so as to prevent hydrogen bond formation. It is due to the stabilizing energy of the hydrogen bond will be lost during folding and unfolding. The amino acid residues that are present in the interior of the protein molecule direct the protein folding. Protein folding decreases its free energy. Energy landscape model. The thermodynamic study of the protein stability can be explained by the energy landscape model. In this model, the protein's energy is considered to be a function of topological arrangement of its atoms. It is a spatial surface having a number of different energy values and coordinates which are separated by ridges and mountains. Every value of the surfaces represents the protein having specific conformation with an energy landscape for each conformation of the protein. Protein denaturation. Denaturation of protein means loss of protein structure due to unsuitable physiochemical conditions. Proteins have its optimum conditions and when kept in unfavorable conditions such as low or high pH, higher concentration of salts, high temperature and high pressure conditions, it loses its native structure and functions, which is also known as denaturation. After denaturation, proteins also do not have defined structure and form aggregates. We have now come to the end of this lecture. Thank you for being there with us. Let us summarize briefly what we have learnt today in this lecture. Protein is a polymer of amino acids arranged in a linear chain. Polymerization of amino acids takes place via the peptide bond formation. Proteins can be classified based on the number of peptide chains, based on composition and also based on their tertiary structure, complexity and number of subunits. The biosynthesis of proteins takes place by the process of translation. Protein synthesis can also be done chemically by peptide synthesis methods. The chemical synthesis methods, however, has some drawbacks. Proteins have 4 levels of structures, namely primary, secondary, tertiary and quaternary. Various techniques like X-ray crystallography, NMR spectroscopy, circular dichroism, cryo-electron microscopy, dual polarization interferometry can be used to determine the protein structure. Proteins have cellular and structural functions. They also perform biocatalytic functions in the form of enzymes. Post-translational modifications of proteins are of 2 major types. One in which the cleavage of bonds of peptide backbone takes place and the other one in which addition or removal of functional group occurs in the side chain of the constituent amino acids. Protein folding plays a very important role in proper functioning of the proteins. Proper folding of the protein is needed for the regulation of the cellular machinery. The 3-dimensional functionally active structure arises due to the particular amino acid sequences in the polypeptide chains when they get folded properly. Denaturation of proteins means loss of protein structure due to unsuitable physiochemical conditions. Thank you for watching this lecture on proteins.